Hello, and welcome to our senior design presentation for the Pfizer-BioNTech COVID-19 vaccine manufacturing process. That was a mouthful. Presenting to you today will be Salim Jalaluddin, Jalen Voles, Andrew Stafford, and myself, Gal Schuster. We will be focusing our presentation on several aspects of the research conducted over this past year. Firstly, we'll give you an introduction. Secondly, Jalen and I will present to you the block flow diagram. Thirdly, Andrew will present to you some of the calculations that were performed, but not limited to the mass and energy balances. And for the finale, Salim will present the costs associated with the process and conclude the presentation with further recommendations. The objective of this project was to produce 1 billion doses of a COVID-19 vaccine with a project life of 1 year. The criteria the team focused on was emphasizing accessibility for all populations of all demographics, while also attaining profitability. The constraints were as follows. The team was limited to 2 semesters and obviously weren't able to get the necessary chemicals and equipment and run human trials. And obviously there was also limited access to the true production process because that is proprietary information. Now moving on to the first part of a block flow diagram. The process begins with plasmid DNA and enzymes that are sent into an mRNA bioreactor, which separates the mRNA from the plasmid DNA with the help of enzymes, all of which are sent into an ultrafilter, which has buffer solution consisting of sodium chloride, sucrose, and water that is sent into it to filter out the plasmid DNA and send the enzymes and mRNA onwards in the process into the chromatography column. Additional buffer solution is sent into the column to separate the enzymes from the mRNA, which will be sent into the capping enzyme reactor. Now to cap off my part of the presentation, I'll be handing it off to Jalen to cover the second half of a block flow diagram. Thank you.
Thank you, Gal. Now for the second half of the process. We start with the mRNA in the capping reactor, which is a jacketed reactor at 37 degrees Celsius. We then send the capping enzymes to start the reaction. This reaction will cap the mRNA, making it readable by the body. After the mRNA is capped, the enzymes and mRNA are sent to a sterile filter, where the excess enzymes are removed and we are left with 900 grams of pure mRNA. The mRNA is then sent to a cross-flow mixer where it is mixed with lipid nanoparticles in order to encapsulate the mRNA. These lipid nanoparticles, or LNPs for short, are made by mixing water and ethanol with 4 different lipids. We now have the encapsulated mRNA and will undergo one last filtration to remove any water, ethanol, or partially encapsulated mRNA. The products of the filter are sent to a storage tank, where they are placed in a buffer solution of multiple salts and sucrose. In this storage tank, we have our finished product, but we must send it to a freezer for distribution. Our final product will contain 900 grams of mRNA, 23,100 grams of LNP, and the buffer solution. Moving on to the process flow diagram. This is going to wrap up the process that Gal and I had discussed into one condensed diagram showing all of our equipment as well as labeled stream numbers. The information for these labeled stream numbers can be found in our mass and energy balances, which Andrew will go over next.
Thank you, Jalen. I will now be covering the mass balances for our plant. So, as you can see on the left-hand side of the table, the streams are numbered according to the stream numbering on the process flow diagram from the previous slide. We were able to construct our mass balances based on the ingredients located in the final product of the vaccine, which we obtained from the emergency use authorization issued by the FDA. Starting from stream 18, which is at the end of our process, we know that each COVID-19 vaccine contains 30 micrograms of modified mRNA and several other ingredients. This stream table as a whole contains the mass for each batch. In our case, one batch is 30 million vaccine doses, which we predicted based on the total number of vaccines Pfizer said they will produce in the year 2021, which was 1 billion doses. Knowing that each batch will take 7 to 10 days, we were able to arrive to the conclusion that each batch could contain roughly 30 million doses. Therefore, each batch will contain 900 grams of mRNA, as well as all of the other active ingredients. Through research and reading through the literature, we were able to understand how they manufactured the lipid nanoparticles, as well as the buffer solution for the vaccine. Working backwards, we calculated the total mass for all of the rest of the components in the vaccine. Moving on to the energy balances, we have 3 primary sources of energy consumption. That is the pumps, one pump corresponding to each stream in our process, the 2 different reactors that we are using in our process, and lastly, the freezers. As you can see in the bottom right-hand corner, we're able to calculate our total energy consumption, which came out to 23,173 megajoules per batch, and for a total time of 3.64 days. Now, this time is just the time that mass will be moving through the pumps, as well as the time spent in the reactor. This does not cover the time that will be spent going through the filters and the columns, as these parts do not contain any moving components. Lastly, we found that we had a total power consumption of 73.624 kilowatts per batch.
Thank you, Andrew. Now I'll be going over the costing that went into our project, first starting with a rundown of the equipment costs. If you look at the second column of the following table, the quantity of each equipment needed is listed. Much of the equipment that we used was single-use or disposable, which is common in the pharmaceutical industry, such as the reactors and pumps. For example, for reactors 1 and 2, we will be needing 34 of each for a project life of 1 year. This is because we will be producing 1 billion doses within 1 year, and each batch takes about 10 days to complete. So 34 batches will require 34 reactors. Another point to consider are the pump types and how both single and multiple use pumps were utilized within our manufacturing process. For example, for pump type 1, we used a single use pump that we determined would best perform the function required, and 270 of these pumps were needed within our process because 8 of these pumps were present and we need 34 batches to produce a total of 1 billion doses. Pump types 2 and 3 are multiple use, so the quantity required for those is less. Most of the equipment we will be using is single use, with the exception of freezers, which will store the vaccine doses, as well as the storage tanks throughout our process, which will vary in size. After costing each equipment thoroughly, we determined that the total equipment cost will be around $21 million. The total capital investment also came out to be around $80 million. I will now be going over the material costs, or the total cost of the raw materials that eventually come together to make the vaccine. FDA-derived materials such as water, sucrose, NaCl, ligand, enzymes, and lipids were all used and the respective costs calculated. We again used 34 batches as our multiplying factor for the quantity we needed for the materials and multiplied this by the cost per unit to get the total costs. One material that stands out in particular in terms of its proportion in the total costing of materials is lipid 2 or DSMC. Around $70 million will be required from just this material alone. In total, the material cost will be around $133 million, and combined with the equipment costs, a total cost of about $213 million will be needed for a process. Now I'll pass it to Andrew, who will be going over the safety regulations that were taken into consideration.
Process safety is a very important consideration to take into account when designing a manufacturing plant, which is why we wanted to highlight it below. The Occupational Safety and Health Administration, or OSHA, along with the Environmental Protective Agency, or the EPA, sets a strict set of rules and guidelines for industry and manufacturing, which must be followed. Some of these regulations that we felt were more directed towards our project have been highlighted below. Now, I'm going to pass it on to Salim, who is going to finish the presentation with our concluding thoughts and recommendations.
To conclude, one recommendation that we would make is based on the benefit that the vaccine provides to the overall greater health of the public, along with the projected profit from vaccine manufacturing. It would be recommended that Pfizer and BioNTech invest in expanding their vaccine formulation operation.